Hello everyone, I am Anuradha. I am a PhD student from School of Computing at the Australian National University. In this talk, I will be talking about how we can adapt plasmid prediction for error-prone long reads. Plasmids are genetic material that coexist with chromosomal DNA. These affect how a microorganism function and behave in their environments. Their study is important because they play a major role in antibiotic resistance and other environmental adaptations. There are two main approaches we can use for plasmid recovery. First approach is to use a reference database. In this approach, we search sequences against the reference database using a tool like BLAST. This approach is computationally expensive and requires a complete database of all the possible species. The second approach is the reference-free approach. In the reference-free approach, we try to use a machine learning classifier to learn plasmid features. Later, we can use these classifier models to classify unseen sequences without using any reference database. Reference-free methods are popular as they provide more generalized approaches for plasmid detection. In our experiments, we consider reference-free methods as our baselines. We can think of two kinds of information for plasmid detection. First, we have the composition information. This is derived by recording the frequency of short substrings called kmers. The frequencies of a given kmer can be used as a vector to represent the composition of a species. Next, we have coverage information. Coverage of a sequence is the average number of times a particular sequence segment appears in a dataset. This is an important indication of the level of presence of a species in a dataset. Coverage information is typically computed by overlapping reads on top of the assembled contigs. We can see several challenges when we move from contigs to long reads. The contigs produced in assemblies are much accurate compared to error-prone long reads. Also, we can derive coverage of contigs easily by using the assembly information or by aligning reads to the contigs. However, we do not have any coverage information for raw reads. Finally, there are plenty of tools to process contigs and extract information such as genes. But we can't say the same for long reads due to their erroneous nature. For plasmid classification, we can use several observations. Sequencing reads from the same origin should have similar coverage and composition. However, it might not be the case for reads from different origins. If we think about a chromosome and its plasmids, they could have slightly different composition. At the same time, their coverages can be significantly different due to varying copy numbers. Usually, we can expect a higher coverage for plasmids compared to their chromosomes. Now we know that we can use coverage and composition to assist plasmid detection. This brings us to our first challenge of computing coverage for long reads. Naively, we can do all versus all alignment. That's millions of alignments which can take time and resources. So, how can we estimate coverage without having to do all versus all alignment? If we consider a read and its kmers, we have accurate kmers and we have erroneous kmers. In our approach, we use accurate kmers of reads to estimate the coverage of the read's origin. Now, if we consider all the reads of the dataset, accurate kmers are more likely to appear across the entire dataset. However, the likelihood of appearing the same erroneous kmer multiple times is relatively low. So, if we consider all the kmers from a read and consider the frequency of that kmer from the whole dataset, we can plot a histogram. In this histogram, we have two peaks for erroneous kmers and accurate kmers. We treat this histogram as a vector to represent coverage of a single read. If we consider multiple reads from the same origin, we can obtain a band as shown in this figure. We can clearly see that all the reads show a similar pattern. We call these resulting histograms as schema coverage histograms. In our tool, we use these schema coverage histograms to represent coverage of a single read. We observe for relatively short schemas, the oligonucleotide frequency vectors are very similar for long contigs and long reads from the same origin. Therefore, we can confidently rely on schema frequencies of long reads for composition information. In our experiments, we use trimer frequencies as vectors to represent the compositions of long reads. The last challenge is combining the coverage and composition information to improve plasmid detection results of error-prone long reads. Existing tools perform well on assembled contigs, and they are based on classifiers trained using composition information from a reference database of plasmids and chromosomes. However, the supervised learning approach cannot use any coverage information as it is not available during the training phase. Two such tools are PlasClass and PlasFlow. These tools show promising results in plasmid recovery of contigs. Although long reads classification is challenging, we still have a small amount of correct plasmid classifications from existing tools. In this diagram, we show the two-dimensional UMAP embeddings of error-prone long reads using coverage and composition. For a very simple example, we can see that there is a visible separation of the two classes. This also means that if we can use these embeddings with high confidence labels, we might be able to classify the unlabeled reads from existing plasmid classification tools. For a dataset with several plasmids and chromosomes, a 2D UMAP embedding would look like this. Even in such a complex dataset, we can still see clear neighborhoods of plasmids and chromosomes. Let's see how we use this ideology in our PlasLR pipeline to improve plasmid predictions. This is the overall pipeline of our tool. In the coming slides, I will explain each step in detail. As the first step in our pipeline, we compute representative vectors for coverage and composition. In our experiments, we use trimer frequency vectors which have 32 dimensions as composition vectors. Our schema coverage histograms has 32 dimensions by default, capturing coverages up to 320x. The resulting vector has 64 dimensions representing coverage and composition. Next, we obtain a lower dimensional representation using UMAP, TSNE, or PCA. This helps us to localize plasmid and chromosomal reads according to their coverage and composition. At this stage, we will have an embedding such that the reads from the same origin are closer and reads from different origins are away from each other. In our experiments, we observed that existing plasmid detection tools can still work on a small subset of reads accurately. So we go ahead and classify the reads using an existing tool. Now we pick the most confident labels and annotate the reads accordingly. At the end of this step, we are left with a large set of unclassified reads with a small set of classified reads. Let's see how we can use the labeled reads to classify the other reads. Intuitively, chromosomal reads and plasmid reads should have consistent neighborhoods. This means we can use the embedding information to rectify any wrongly labeled data points. For this, we observe the neighborhoods for ambiguous labels. We remove such labels and mark those points as unclassified. Now, using the remaining labeled reads and a nearest neighbor classifier, we classify the remaining set of reads. Nearest neighbor classifier computes class probabilities for all the unclassified data points. In our experiments, we use points with probability above 0.5 as plasmids and the rest as chromosomes. We used several simulated and real datasets in our experiments. We used SimLoad with default parameters for simulating PacBio reads. We used the SIMO Merck community dataset to obtain real plasmid reads. We vary the decision probability threshold and plot the precision recall curve to see the behavior of each classifier. Here you can see the precision recall plots for a dataset in our paper. This shows that we can improve the plasmid classification by incorporating dataset specific information such as coverage. This table shows the improvements to plasmid classification using PlasLR on top of the two common plasmid detection tools. We can see that we attained significant improvements in classification by incorporating coverage and composition neighborhood information. Classification of reads before assembly allows us to evaluate the potential impact on final complete assemblies. First, we separated reads based on the class predicted by our tool. We assembled these reads separately using MetaFly. We used MetaQuast to compute the genome fraction and the number of reference sequences recovered in assembled contigs. These results indicate that plasmid separation before assembly improved the overall quality of resulting contigs. This also means that some plasmids may never be recovered without a pre-assembly separation process. Sometimes we even improve the chromosome assemblies. This is because by separation of plasmids, we allow assemblers to estimate better internal parameters. In conclusion, we showed that we can use unsupervised embeddings to improve results from supervised learning approaches. As a result, we were able to use existing tools without any modification. Finally, we got to improve the assemblies that would be very helpful for downstream analysis. In future, we intend to extend our approach by combining binning and plasmid recovery to classify each plasmid and chromosome separately. Finally, I would like to thank my collaborators, Ms. Vichini and Dr. Yulin from ANU Computational Genomics Group. Also, I thank Dr. Liagrun Pu from Tel Aviv University for her support in this work. Thank you all for joining my presentation. I am happy to answer if you have any questions.